Hi there. In this video, I'll be discussing genetic recombination, or simply recombination. As I stated in the last video, it's one part of DNA repair, but more importantly, it's a major source of genetic variation in organisms. So people commonly think of genetic recombination during meiosis, which is on the far right here, so you can get crossovers between all four chromatids during prophase in meiosis. And all four chromatids, of course, are brought together during meiosis. Crossovers can also occur during mitotic division, so you can get crossovers between the sister chromatids here, but you can also get crossovers between homologs. So there are crossovers that occur rarely, but they do occur between the homologs as well. This usually occurs much more readily in regions that have a lot of repetitive elements. So the chromosomes can line up and misalign with regard to the number of repetitive elements, but they still align within a repetitive element. So one chromosome in each case then receives more of the repetitive elements, and one has fewer of the repetitive elements after the crossover is resolved. This is one of the major ways that leads to variation in ribosomal RNA gene variability in plants and animals, where you get unequal crossovers in somatic cells that cause differences in the number of tandem repeats of those ribosomal RNA genes. And I talked about this in detail in one of the previous videos. Even when the chromosomes are not duplicating and have not been duplicated, so this would be in the G1 phase or in early S phase, you can still get crossovers between the homologous chromosomes. And again, this happens more often in repetitive regions. You can also get crossovers between non-homologous chromosomes. However, in most cases, that results in some detrimental effects in the cell, including many of these that are involved in tumorigenesis and carcinogenesis, or cancer. Here I want to discuss a few of the details about crossovers and genetic recombination. So if you have two chromosomes lined up, and they can be bacterial chromosomes, eukaryotic chromosomes, anything, and on one, this blue DNA fragment here, I have the dominant alleles. They could be anything. And on the red chromosome down here, I have the recessive alleles, just as markers, so you can see where things are going. The recombination event is initiated by first lining up, and then through a single strand break that's usually accomplished with an enzyme that breaks in one place. And then single-stranded binding proteins are recruited to the area to stabilize the single-stranded regions that result from that breakage. This is followed by single-strand invasion by this red strand into the blue strand there. And this displaces one of the strands of the blue chromosome, so that now you have a partial crossover created in this case. And you can see here that the single-stranded regions are stabilized by the single-strand binding proteins. Because this one single-strand region of the blue chromosome is complementary to that on the red chromosome, then they can form what's called a heteroduplex. So now it has partly the red chromosome, one strand is the red chromosome, and one strand is from the blue chromosome. So next, on this side of the molecule, you have completion of that partial crossover, so then you get DNA ligase on both strands, but you still have a crossover in the middle here, a true crossover now with between the red strands and the blue strands. And if you stretch that out in a different way to concentrate on the crossover region, then you have this top part with the dominant A allele, a heteroduplex region of the B allele, and then the dominant D allele. And on the other strand, or the other DNA part of the DNA molecule, you have the recessive A allele, a heteroduplex B allele, and the recessive D allele. There are two different ways that these are resolved. So here again is that same structure up here. If you cut across here and then ligate, what you get is this structure here. So you get the two chromosomes resolved at the crossover. So you have one with the dominant A allele, and it has the dominant D allele. So the overall chromosome has not really changed ends. But then you've got this heteroduplex B allele. On the other chromosome, the red one, you have the two recessive alleles for A and D, and then a heteroduplex, sort of the complementary heteroduplex of the B, large B, small b allele. And I'll be talking about that heteroduplex region shortly. We saw that before also in another case in our recombination-dependent replication, when I was talking about DNA replication 2 videos ago. So these are the same types of heteroduplex regions that were produced in that reaction also. So there's another way to resolve this crossover also. If you just rotate this bottom section here, move the small d over here and the small a over here, rotate that, it opens up the center here called a chi structure. And then if you rotate this over here, exchanging that small a there to over to here and the large D over to here, now you have a crossover in the center here. And you might ask, does the DNA really do that? And yes, actually DNA actually does this. The DNA strands in a crossover are closely connected, and they twist and turn with respect to one another, with the help of proteins, by the way. So that's also, those are also involved here. So now when you cut and ligate across here, you get different molecules than over here. So now the resulting chromosomes look like this, where on one chromosome you have the large or the dominant A allele and the recessive D allele and a heteroduplex B region. And you have on the other chromosome the recessive A allele, the dominant D allele, and a heteroduplex region for the B allele. And in this case, you could look at that genetically and otherwise and say a crossover did occur there, whereas in this case, over here, it's difficult to discern that a crossover did occur in that region, even though, yes, it had occurred. One way that you can tell is by looking at these heteroduplex regions. In addition to finding these in bacteria, they were studied quite a bit in ascomycetes. Specifically, this is in Neurospora crassa. So a couple of videos ago, I talked about ascomycetes and how they form these asci, or this is an ascus, where there are 8 ascospores. So you can do genetic crosses and then take the spores out using a dissecting scope one by one, and then you can grow each one of those spores and look at the genotypes and the phenotypes of those spores, those individual spores. So you can detect how different alleles are segregating within that population of cells. When this was done, these crosses were done where you had one strain that was the wild type and then one strain that was the mutant type. And then people would go through and look at the asci, grow those on media that would show a difference between the wild type and the mutant. And what you would expect is this, a 4-to-4 segregation of those haploids within the ascus. So there would be 4 of the wild type and 4 of the mutants. But what they found was, in some cases, there was a 6-2 or 2-6 segregation. So the spores, there were 6 that were wild type and 2 were mutant, or 6 were mutant and 2 were wild type. And they even had some that were 5-3 and 3-5, and some that were 3-1 to 1-3, in that they were arranged differently within the ascus. And it's a very regular segregation of the spores in the ascus. So something different was happening. And what was happening was that there was gene conversion or chromatid conversion. And this always happened in an area of a crossover in that heteroduplex region. So in the heteroduplex region, you have one half of that chromosome that would be the wild type, and the other half would be the mutant. And in about equal numbers of times, the repair mechanisms would go in there and correct any of those base pairs where they weren't common base pairs, where there were mutations. But in some cases, it would repair one strand more than the other strand. So instead of getting a 4-4 segregation, you would get different proportions of segregation of those two alleles, primarily because of conversion of those strands to the other type by using the other strand as the template. So in many cases, you would get this normal 4-4 segregation because the correction was on one strand half of the time and on the other strand the other half of the time. So you still got a 4-4 segregation, as if there were no crossover in that area. However, in some cases, one strand was used more for the correction template than the other, and then you would get varying numbers of the wild type allele versus the mutant allele. And even on very rare cases, you would get 8-0 segregation of those, meaning that even though you had put in two different genotypes, you got only one genotype out on the other end, either the wild type or the mutant type. 8-0 or 0-8. Okay, now I'd like to talk about a little more about recombination in some specific examples of where it's functional and important to different parts of living organisms. And the first one is the obvious one, that is in meiosis. So you get a genetic recombination to form more and more combinations of the various genes on each one of those chromosomes. So you have these recombination events creating new combinations of those genes that creates a large number of gametes that can join with the other gamete from the other sex. And create new combinations of genes, many of which will contain deleterious genes. And so those zygotes formed from the joining of those two chromosomes will probably just die. But it also creates the possibility for some combinations of genes that are definitely advantageous to that organism. And so that sort of allows certain gene combinations to survive. It also has allowed our genomes to get very large. If we didn't have the process of sexual reproduction, then our genomes would be very much smaller, and evolution would be slower. The only way to have these very large genomes with a large number of genes is to have this sexual reproduction where you recombine genes, hopefully, and in many cases, creating new advantageous combinations, but also getting rid of deleterious combinations. And this often happens just in gametogenesis, that those eggs don't survive very long because they already have deleterious mutations, and so those are taken out of any possibility of formation of a zygote. So that has been a major driving force in evolution of organisms, multicellular organisms with very large genomes, such as us and a lot of protists and plants and other multicellular organisms. Without recombination and sexual reproduction, we just would not be here at all, or even close to being here. However, there are some problems with recombination. One of them is that you can produce large deletions, which can be deleterious or even fatal to that cell and to that organism. Often the recombination events not only cause a deletion, but they cause a duplication. Duplications can sometimes be deleterious, sometimes be beneficial. So this one is sort of a wash as being plus or minus. You can also get chromosomal inversions that can affect expression of some genes also, either in a positive or a negative way. And then in some cases you can have chromosomal translocations. So down here you have a translocation between chromosome 4 and chromosome 20. So now you have a shortened chromosome 4 with a piece added on from chromosome 20, and a chromosome 20 that's longer because it has a long piece of chromosome 4 attached to it. And here's one of those. Here's another one where you have a chromosomal translocation between chromosome 12 and chromosome 17, where smaller pieces are exchanged, and both of these cause cancers. Specifically, this one, chromosome 8 and chromosome 14, exchanges between those two cause a type of B lymphocyte tumor called Burkitt's lymphoma. So this exchanges two very important gene portions that causes a failure of function of one of those that leads to tumor formation and cancer. Here's a map of the 23 chromosomes of humans and showing some of the crossovers that lead to different types of genetic disorders, including this one between chromosome 8 and 14, which cause that Burkitt's lymphoma. 14 and 11, a crossover there, causes this mantle cell lymphoma. There's a crossover between 14 and 18, causes a follicular lymphoma. And then there are other ones here, Ewing's sarcoma between chromosome 22 and chromosome 11, and many others. You can see there are other diseases such as schizophrenia here and Down syndrome being caused by chromosomal translocations. And that one's between chromosome 14 and 21. Most of you think of Down syndrome as being trisomy of 21, but Down syndrome can be caused by chromosomal translocations, and all of those also involve chromosome 21. Many of these cause fusion genes, that is, the crossover occurs between two genes, usually in an intron region. That causes a fusion transcript and then a fusion protein. And many of those cause increased cell proliferation, decrease in apoptosis or cell death, decrease in differentiation, and increase in motility. And so most of those are characteristic of cancer cells. So in this diagram, it sort of expands on that idea in that you can get this fusion gene, a fusion RNA transcript, and then a fusion protein, which changes its function. Usually it loses function, sometimes it gains function. You can also get it by this enforced expression of an oncogene. So normally certain genes are turned on to a limited extent. But if they're placed next to a very active promoter, such as in this case or this case, then the promoter can cause that gene to be expressed and the protein to be expressed in much higher levels than normal, and that will change the function of that cell and often turn it into a cancer cell. So that's just one type of change that can lead to cancer. So you can see In this case, the acute myeloid leukemia, about 20% of those cancers are caused by gene fusion. In chronic myeloid leukemia, another blood cell cancer, 100% of those are caused by gene fusions and then protein fusions. And then down here, a couple of other ones, acute lymphoblastic leukemia, mature B cell neoplasms, and mature T cell neoplasms often are caused by gene fusions. And then another one down here at 80% are male genital organ cancers caused by cell fusions. And a couple others down here, soft tissue and some bone cancers, are caused by chromosomal translocations that cause gene fusions. Recombination events often cause changes in cell surface proteins among pathogens, and this one's for a trypanosome, but it also occurs in the malarial parasite and other parasites, in that the immune system often reacts towards particular cell surface proteins of pathogens, and in many cases they're on the flagella. So the flagella have certain cell surface proteins on them, and they're recognized eventually by the immune system, and it kills that pathogen. But so pathogens have evolved to switch those cell surface proteins, and they do this through recombination events normally. So on one chromosome you have a variable region that can be switched out for other variable regions on those genes, and then that forms a protein that then changes with regard to the epitope or the type of protein that's being put out to the cell surface. So this confuses the immune system. The immune system is first reacting to one protein, and it kills all of the pathogens with that, but then there's a crossover event in some of those pathogens, and then they're able to change the cell surface protein, first the gene and then the cell surface protein, which is not recognized by the immune system until the immune system finally then recognizes that protein and starts killing that one. That's one reason why some of these pathogens stay in the body for quite a while. So it looks like people sort of get sick, and then they tend to get better, and then they get sick again, and then they get better, and then they get sick again. So that's because the parasite is able to change the proteins on its cell surface, so the immune system can't catch up with those changes for a while. It actually has three different modes of switching. The first is called gene conversion, and this is a recombination event where it switches out one of these variable regions into the expressed region for that gene, where it's then expressed as the RNA and then the protein. So it switches each one of these little variable sections into that site, so it becomes part of that gene, part of the protein. Another one is via telomere exchange, so that's also a crossover event where you're doing a crossover and exchanging one piece of one chromosome with another piece of that chromosome. And the third one is a transcriptional switch, and this is usually not involving a crossover event, but just changing which version is expressed at a particular time. And then there's recombination involved in formation of immunoglobulins in the T and B cells. There is a gene for the heavy chains and a gene for the light chains, and then various ways to attach those together to form various types of immunoglobulins. And so they have a constant region here in lighter blue, and then this orange region out here. Those are variable regions, and so this protein is formed from genes that have many, many different variable parts. So to form this antibody on the bottom here, it contains these constant regions in blue, and then these variable regions in red, green, and yellow. And in the gene, on the chromosome, it's set up this way. There are many variable red sections, many D segments, many J segments, and 3 or 4 or more constant regions. So early in the maturation of that cell, you get recombination between the D and the J segments. So now, and that's in the DNA, so now the DNA has changed. It won't change back. It's lost that central section of that gene. And then later on, there's another recombination event, choosing just one of the D segments and retaining 3 of the J segments and the constant regions, but then selecting out only a few of the variable regions. And so once that's transcribed, then it becomes 1 red, 1 green, 1 yellow, and then the 3 blue segments to create that protein, that RNA, and then the immunoglobulin itself. And here's a slightly different version of the same thing, showing that there's 150 genes in 15 families, about 2.5 million base pairs long. There are at least 12 versions of the D regions and 4 versions of the J region, and then 8 versions of the constant regions. And so the first recombination event is early in development, and then a second recombination event later in development. And all of these other regions disappear from the genome. They're no longer in the genome. And then finally, you transcribe this and it becomes the immunoglobulins. The constant regions stick into the membrane of the host cell, and the other regions stick out and then interact with antigens or the antigens on the pathogens. And the B cells do 2 things. One is they present those antigens that they've collected to the T cells to tell them, to communicate with them. But they also release some of those antibodies into the blood system and other fluids. So that kind of soaks up other antigens that happen to be floating around, antigens of the specific type that interact with that variable region. I'll discuss this in a little more detail when I have the video on the immune system. So here's a development of those B cells, B memory and B plasma cells. So you have a proto-B cell with the complete genome, and then a pre-B cell here, type 1, where you're getting recombination between that D and J region. So you're losing a little part of the genome here, and then you get rearrangement of the variable and the D and the J regions. So now you've lost a little more of the genome, and then they start maturing and producing those immunoglobulins, and eventually you have a naive B cell that then can react with the antigens, and if that particular B cell produces a variable piece that interacts with the J cell, then the T cell will initiate proliferation of that particular B cell and signal other parts of the immune system also to react to that antigen. So again, recombination is intimately involved here, and so that once a B cell is mature, most of the gene region for both the heavy chains and the light chains, which both undergo recombination, much of those gene sections are gone. So that B cell eventually can only form one type of immunoglobulin. It can't go back and try to form different types. Those parts of the genome are completely gone. Here is sort of a summary of some of those changes that I've talked about already. One is crossing over, so you can get a crossover in a chromosome, thereby producing a couple of new types of chromosomes upon duplication. One contains sort of a hybrid of the other two that were originally there, and one is just like the original one. Secondly, you can get gene conversion by converting one form into another. Sometimes that's done by recombination. Other times it's done just by replication, by putting these two chromosomes together, creating sort of a heteroduplex region, as we've talked about before, and then one form gets copied over the other one, so that now you have two of the alpha alleles here rather than an A and an alpha. And then a third part here is an entire chromosome can be deleted and the other one can be replicated, so that you still have two chromosomes, two homologous chromosomes in that cell, but one has been completely eliminated and the other one has been copied in its place. So then to fill out this table, then we've talked about mutagens and how those can create mutations, replication errors, altered base pairing, and repair enzyme pathways. But then I talked about other types of changes in the genome, including gene rearrangements. Many of these involve recombination events, including chromosomal translocations, and these can also create heteroduplex regions that lead to the gene conversion of one gene section over another. And then finally, you can have whole chromosome deletions and then duplications of the alternate homolog. So a lot of different changes can occur in the genomes of organisms. And one final one that actually happens fairly frequently is polymerase slippage, and this often happens in regions where you have a lot of small tandem repeats. In this case, you have a lot of tandem repeats. The polymerase slips off a bit from the template, or there's a release of one part of the template, and then it folds back on itself. Since all of these are self-complementary, they can bind in many different ways, and then you can create a region that has more of those repeats or fewer of those repeats, depending on how that polymerase slips. Now, this can be in small repeats or it can be in large repeats, although it's more common with small repeats because they're so close together with respect to the DNA polymerase and attendant molecules. So this is the end of this section that included two videos going from DNA damage, that is mutation of various sorts, including those from chemicals, from bacteria, from replication systems, from repair systems. And then I talked about some of the ways that DNA could be repaired and other ways in which it could be both repaired and damaged, sometimes at the same time. Okay, so that's the end of this video. That was primarily about genetic recombination and recombination in general, that can be used to create diversity in genotypes, as well as diversity in cell surface proteins in the case of pathogens, and it can also lead to chromosomal translocations that can cause various diseases, including cancer. Okay, well, thanks for listening and viewing, and I will talk to you later.